Welcome to the Scientific Sense podcast, where we explore emerging ideas from science, policy, economics, and technology. My name is Gill Eapen. We talk with world's leading academics and experts about their recent research or general areas of topical interest. Scientific Sense is an unstructured conversation with no agenda or preparation. We cover a wide variety of domains where new discoveries are made and new technologies are developed on a daily basis. We are most interested in how new ideas affect society and help educate the world how to pursue a rewarding and enjoyable life rooted in science, logic, and information. We seek knowledge without boundaries or constraints and provide unedited content of conversations with researchers and leaders who love what they do. A companion blog to this podcast can be found at scientificsense.com, and this podcast is available on over a dozen platforms and directly at scientificsense.net. If you have suggestions for topics, guests, and other ideas, please send them to info@scientificsense.com, and I can be reached at gill@eapen.info. My guest today is Professor Howard Fields, who is Professor Emeritus of Neurology at UCSF. He was a founder of the UCSF Pain Management Center and has made major contributions to understanding and treating neuropathic pain. Welcome, Howard.
Good afternoon.
Yeah, so thanks for doing this. So I want to start with, I know that you are a well-known, one of the world's experts on pain. I want to start one of your older papers from 2006 entitled A Motivation Decision Model of Pain, the Role of Opioids. You say pain is a sensation like touch, vision, and olfaction, and it is defined by its subjective properties. Although activation of primary afferent nociceptors initiates a variety of responses, it is a relation of the noxious stimulus to the psychophysical properties of the subjective experience that has attracted the attention of most researchers. Howard, I used to think that pain is pretty simple. Some electrical impulses travel up the nerves into the brain, and that you feel pain. There is something more to it?
Oh, there's a lot more to it. It is relatively simple compared to, for example, vision or olfaction. We know the detectors in the periphery, we call them nociceptors, which means that they detect actual or impending tissue damage. We know how the information is transmitted. It's just like every other cell in the nervous system through action potentials that are conducted from the tissue that's been threatened or damaged to the central nervous system. We know the pathways, we know the neurotransmitters, we know the cortical areas that are activated by that stimulus. We've even got information now from functional imaging, functional magnetic resonance imaging in awake human beings, where we can correlate cerebral blood flow with reports of pain. And this has given us an understanding of where in the cortex you need neural activity to produce the sensation of pain. So this has been a tremendous set of experiments over the last 30 years or so to get all this information. But there is still at the end a very mysterious process that we don't understand at all. In fact, I would say it's one of the sort of major difficult problems in neuroscience, sensory neuroscience in general, which is how do you convert the electrochemical pulses in the neurons that they use to communicate with other neurons? How does that activity translate into a subjective experience? That's completely mysterious.
And that subjective experience is different for different people. That makes it more complicated, right?
Well, I don't know if I agree with that.
Oh,
It's hard to know what somebody else's subjective experience is, so it'd be
yeah.
difficult to compare it in different people. That said, what I do agree with is if you apply a given painful stimulus to different people, they will report different levels of pain and perhaps different qualities of pain. So it's true that there's a highly variable relationship between the tissue damaging stimulus and what different people report. That's for sure true. Maybe that's what you were driving at.
Yeah, that's what I was driving at. So I remember this was a long time ago when I was at Pfizer. Company was developing some pain medications. And obviously, one of the difficulties in clinical trials in pain is to get a consistent set of responses from the
That's
patients.
correct.
And there's always been the measurement problem has always been a difficult one, right?
Well, you can constrain the situation in individuals, and they can be trained to give very reliable reports. And if you're using a thermal stimulus, so you can accurately control the temperature, there's a fairly reproducible relationship between the intensity of the stimulus and the numerical rating scale. So that you would use something like a 0 to 10 scale, where 0 is no pain, 10 is the worst pain you've ever seen. If you're around 43, 44 degrees, most people say, yes, that's moderately painful. I'd give that a 5. As you go up, they will rate it higher and higher. However, in the real world, it's very variable. You can never predict, what somebody's going to tell you, say after they've, like sprained their ankle or even broken a bone. That in the clinical situation, it is incredibly variable.
Yeah. So I want to go back to the mechanism. And so you hit your leg against something, some sort of tissue damage happens on site. And from that site, some information is sent to the brain, right? That is the mechanism by which the brain knows something bad has happened.
Correct.
And at that point, the brain is converting that information into some what exactly happens in the brain?
Great. Yeah, that's a good question. Well, we know the physics and chemistry of it. We know how nerve cells work. We even know how they talk to each other, right? So every nerve cell pretty much is the same in the sense that it has what we call an action potential. An action potential is a rapid, in other words, anywhere from a half a millisecond to 2 or 3 milliseconds. You have an inward rush of sodium, and you have then an outward rush of potassium. So you have a depolarization and a repolarization. We call that the action potential, and that's the same in all neurons. And so then that action potential is conducted down the axon to what we call the axon terminals, and it depolarizes the terminals, and those terminals release a chemical signal which goes across a gap called the synapse and then acts on the next cell. It could be either an excitatory or an inhibitory effect. So what you have with a painful stimulus is you evoke a pattern of excitations and inhibitions in a set of interconnected neurons. And when that set of interconnected neurons fires in a certain way, you get the perception of pain. That's how it happens. Now, as I said before, how that gets converted from the objective chemical changes that we can measure to the subjective experience of pain, that's a complete mystery. So I can't tell you. I can tell you what you need to make it happen, but I can't tell you how it happens.
Right. Yeah. So intuitively, this should have had survival benefits.
Has to,
This
yes.
is sort of helping Homo sapiens survive, so to speak, right? The pain process. Do we know of any biological systems, any animals who don't feel pain?
Yes. Well, this is a really great question, Gill. Can I call you Gill?
Yes.
Okay. So it turns out that there are animals that don't have nerves. Sponges, that whole phylum, they are animals, but they don't have nerves. And if you don't have nerves, then you can't feel pain, right? The most primitive animals that have a nervous system are in the phylum Cnidarian, and those like sea anemones are cnidarians. They don't have a brain, but they have a nerve net, and they can do two things. They can rapidly respond to threats by escaping, or they can rapidly respond to a food source by moving toward it. So those are the two things that it can do. And that illustrates a general principle. You can ask yourself, well, what is the nervous system for? How does it help animals to have a nervous system? It helps them by allowing them to change rapidly and move rapidly in response to rapid changes in the environment that either threaten their survival or are necessary for their survival. So animals need, a source of food to survive. All animals, that's almost the definition of an animal. Plants don't need a source of food. They can make their own food from minerals and sunlight. But animals need to find food, and having a nervous system helps them move quickly to acquire it. And if there's a threat of damage or of destruction, a predator, for example, they need to rapidly escape and respond to that in one way or another. So in order for your sensory systems to help you survive, you have to have a behavioral repertoire that allows you to react in a meaningful way to the particular stimulus, the particular change in your environment.
And so the nervous system formed, as you say, for either to counteract a threat or to get some sort of reward like food.
Correct.
And is it sort of instinctual? So when we touch something hot, when we pull the hand out, is the brain doing it consciously, or is it something instinctually? It's a different process altogether.
Yes, I would say that if it's what we call a reflex is a motor response that's triggered automatically by a particular sensory input. So you don't need to be awake to pull your hand away from a hot, say, pan. You don't need to be conscious of it. In fact, probably most people have had the experience of touching something hot, pulling their hand away, and then only after they pulled their hand away, feeling the burn. So I think if you have to wait till you actually have the subjective experience to react to it, it would be too slow to protect, your hand from a burn. You have to be able to do it quickly. The information doesn't even need to get to your brain. It just needs to get to your spinal cord.
Yeah, so that's what I was going to ask. So somebody in a coma would do that too?
Yes. Most people in a coma will respond to pain. You can be at least slightly anesthetized and still have a pain response. When an anesthesiologist is trying to gauge your depth of anesthesia, what they do is they, raise the concentration of the anesthetic to the point where you just stop responding to pain.
So you talked about the circuit that gets information to the brain. There is, I think there is something else that sort of inhibits that.
So that's a very important point. It's in the paper that we're talking about, the motivation decision model.
Yeah.
So it turns out that the stimulus, let's say a noxious stimulus, that would generally provoke the sensation of pain, you have to remember that it isn't just a neutral stimulus like, say, seeing an apple or smelling, an apple pie. This is a stimulus that carries with it the prediction that if you don't respond to it, you're going to damage, you're going to suffer some damage. There's going to be a cost to you. So the thing that is important to remember about painful stimuli in particular is they're not just about what's happening. They're a prediction about what's going to happen, right? What's happening is, you're near a red hot, iron. The prediction is that if you touch it, you're going to get a burn, you're going to get tissue damage, and that's not going to be good, right? So knowing that, you want to escape as quickly as possible, and you activate your motor system. In addition, because it's predictive, there's what we call a top-down modulatory pathway. We call it the pain modulating system. And this runs from the cortex through the brainstem back down to the spinal cord, and it controls the volume on the pain intensity scale. The point of that is that, generally there's going to be a time course for the pain intensity. And this is a really important point. So I'm going to go over it slowly. If the pain, let's use a temperature because it's the simplest to understand. If you put your hand on something and the temperature is rising, the prediction is going to be if you leave your hand there over the next second or two, it's going to get worse. So you want to pull your hand away. But let's say that it's mildly painful, but instead of the temperature rising, it's falling, right? At that point, you think, oh, well, I don't need to worry. The temperature is going down, so I don't need to respond to it. Now we have evidence that this top-down system can control sensation bidirectionally. If the stimulus intensity is rising, it amplifies the signal, so it feels worse at any given temperature than it would if it was a steady state temperature stimulus. On the other hand, if the temperature is falling at any given temperature, it's felt as less intense than a steady temperature. So, we have evidence from functional imaging in people that this falling temperature engages this top-down inhibitory system. It can also engage a top-down excitatory system. The top-down system is bidirectional. One group of cells amplifies the pain. We call those on cells. There's another group of cells in the brainstem that inhibit pain, and we call those off cells. The off cells are activated by analgesic medications like morphine. They can be activated by endogenous opioids, we call endorphins, and they can produce actual pain relief by suppressing the pain transmission pathway and suppressing it at multiple levels, including at the earliest entry to the nervous system, which is at the spinal cord.
This is so fascinating, Howard. I sometimes when I think about these things, I think about computers.
Exactly.
It seems like a packet of information is sent because it's pretty important that information arrives, and then you have to follow up with that package of information. In this case, sort of a slope if it's temperature, sort of the slope of how the temperature is changing, so that you can do some processing and give some sort of a threat level gauge, so to speak, right? So that the body can react appropriately.
Yes, and I would say that there are some formal similarities between the operation of the nervous system and the operation of a computer. I think the most important one is that the brain operates by moving information around. It operates symbolically. A particular set of connections, a particular set of timings for impulses, a particular amount of chemical released at the terminals is what creates the sensation. That's how it works, right? But if the connections are not correct, it's all nonsense. The brain doesn't work. You have to have the right connections, and you have to have the right temporal pattern of ons, and offs, right? And the brain's a little bit different from most modern computers in that it has series connections between information that's represented digitally and information that's represented in an analog fashion. So the digital representation are the action potentials. The key thing there is the interval between successive action potentials. That's where the information is carried. But at the synapse, it's analog in that you can have a lot of small inputs. They're all added if they're excitatory or inhibited if they're inhibitory at the surface of the second order cell. So it's sort of, I would say, your brain is an analog digital computer hybrid.
And so this gives us as a way to control pain. So this inhibiting channel, if you can influence and affect that, you can reduce pain artificially.
Well, I don't know if I'd use the word artificial. It's a natural biological system.
Yeah, I mean, sort of some external agents going in those channels.
The external agent is the information that the nervous system uses to process, right?
Yeah. I meant so let me ask you though. So the pain inhibition that happens, do we have chemical agents that could moderate?
Yeah, we do.
That?
Those are the opioid analgesics, right? So Percodan, Vicodin, morphine, all of these are clinically used analgesics that work on the system, and they increase the activity of the off cell. They also have direct effects on the spinal cord. So you can put morphine directly on the spinal cord to get an analgesic effect that's local to the area where you put the morphine on, right? So the morphine acts at the same biological target. It's what we call the mu opioid receptor. And these morphine, if you give it, say, intravenously, it will get into the nervous system. It will find those receptors and it will bind to them. Normally, it's endogenous opioids, like enkephalin or beta endorphin, that act at those receptors. So when you give somebody morphine, you're mimicking the effect of the natural, what we call the endogenous opioid.
You have another paper, state-dependent opioid control of pain. So you say that the mu opioid receptor are powerful analgesics and are highly addictive. However, the contribution of the opioid and opioid receptor-like receptors to motivational states is less clear. So how does motivation come into the equation?
Well, as we know, pain itself is a motivation, right? So, and the motivation is, I need to protect myself. I need to protect my tissues. There can be conflicting motivations. So, for example, you may be in a situation where you're hungry, but your back hurts. If you weren't hungry, you just stay in bed and rest, but you are hungry, you're very hungry, and you feel like you have to get up and get something to eat. So there are situations in which pain, let's call that motivation A, is competing with a different motivation. Let's call that hunger, motivation B. So how does the brain decide whether to, go for the food and ignore the pain, or respond to the pain even though you're hungry? So this is sort of the basic conundrum that the brain has to deal with. And there are going to be a lot of variables that enter into that equation. You're computing the cost-benefit relationship of a variety of things that you could do, and keep it simple by just talking about food. So this is a different part of the nervous system. There are places in the forebrain, usually subcortical areas. Again, these areas have the mu opioid receptor. And in animal experiments, if you inject a mu opioid agonist, let's say like morphine, and you activate the mu receptor in these sites, you can shift the animal's bias toward responding to food and at the same time get an analgesic effect, right? So if the decision that you make based on your cost-benefit computation is, I'm really hungry right now, this food tastes really good, I'm just going to go ahead and eat, you get an analgesic effect, right? So there's an analgesic effect of food. The other thing that's very interesting is that it seems like this same neural mechanism is involved in promoting alcohol consumption. So alcohol is present in most ripe fruit. So a lot of primates that are primarily fruit eating have a certain amount of alcohol in their diet. The alcohol indicates that the fruit is ripe, ready to eat, and is energy intense, which is what animals like. So if it looks like when you start to nibble away at some fruit and it's got a little alcohol in it, you get the release of an endogenous opioid in these sites of the brain that are involved in decision making, and that makes you drink more alcohol or eat more of an energy intense food, right? So again, all of these behaviors that we talk about, analgesia, we talk about addiction, we talk about alcoholism, but they're all manifestations of innate biological, motivational states.
Yeah. And so the cost benefit calculation the brain is making in many experiments forward, the one complication is that when you image the brain, you see activity in a lot of different
That's
sites in the
correct.
brain. Is that
Yes,
the case for pain
very
too?
much so. Yeah, there's widespread activity. There's been some very nice what they call meta-analyses, where they've taken, I would say, hundreds or thousands of scans, and if you do enough of them and you have good psychophysical data, so you're asking people, what is the intensity of the pain you feel, and you're gradually changing the stimulus intensity to match it across different people, you can pick out what they call the neurological pain signature. That isn't anything I've written about, but it's you can Google it if you're interested. There are specific, relatively limited areas that show a very tight correlation with pain intensity. There are other areas that show activity, but they're not specifically highly correlated with stimulus intensity. One of the interesting findings, which I really like, but which is a bit of a head scratcher, is that there's a slightly different distribution of the sites in the brain that correlate with stimulus intensity. And then there's a slightly different set of sites that correlate with people's subjective report, right? So there's not an overlap of the areas they're activated by the stimulus and the areas that are activated by the pain report. And that makes sense, thinking about what I just said, in that it's not simply pain is more than a sensation, right? It's a motivational state.
Yeah.
And what you report in terms of intensity is going to depend on the history of the stimulus, not just the intensity at the time of your pain rating, right? So there's inherent nonlinearities in the system.
Yeah. No, I was also thinking about this decision-making process you talk about. So if different parts of the brain is involved, I wonder, is some sort of a voting mechanism in place? Somebody has to make a decision. If 3-4 places in the brain you have data, you do some analysis, and as you say, the intensity report, some of it is objective intensity is measured somewhere, the subjective valuation is done elsewhere. There has to be some sort of a voting mechanism ultimately to say, pull the hand off right now.
And that's what I would say, that's inherent in the wiring. And in addition, you need to have a place in the brain where you have what I would call a kind of currency, where you can, use the same currency to weigh the cost of tissue damage against the value of eating a certain amount of food, right? Well, the nice thing about the nervous system is everything is translated into neural activity, right? So you don't really have a big engineering problem. You don't have to deal with, changing the modality. Everything is in neural activity. So that solves the biggest problem right there, right? So then you could ask the question, are there places in the brain where you see neurons that respond in one direction to costs and in another direction to benefits? And there's a couple places in the brain that do that. So you'll have a convergence of the inputs that are, say, computing the value of a reward. You'll have another set of neurons that are computing the cost of a painful stimulus in terms of potential tissue damage, and they converge, onto a single neuron that's, say, excited by reward and inhibited by pain. So that would be, there's some evidence that a set of neurons in the midbrain, an area called the ventral tegmental area, which has dopamine neurons. You may have heard of dopamine. It's considered by many to be a critical neurotransmitter for encoding reinforcement or reward. And there's so these same neurons that are excited by reward are inhibited by painful inputs.
Right. But the cost benefit, sort of decision is still pretty complex, right? So going back to the heat example, there is sort of a short-term cost. And then if you're contrasting that with the requirement of food, there is long-term cost. And both of these are sort of dependent on the state of the individual, right? If the individual is, for instance, close to death without food, that's a different decision. So I just wondered, even though there is a common currency there in terms of neural activity, it is still a very, very complex trade-off decision the brain has to make.
Yeah, I have to think about that. What I would say is that it's an equation with multiple variables, and so by that definition, you could use the word complex, but there are things that are really complicated, So, for example, let's talk about dreaming or imagining or writing poetry or composing music. Those are things, to a certain extent, we do not understand at all. And they certainly don't make a whole lot of sense in terms of either a cost or a benefit, right? Whereas food, you can do calculations about what number of calories you need to sustain your body weight, and you can do calculations on how much damage a stimulus of a given temperature can produce. And that's something that I would think over the course of 400 million years of evolution of the nervous system, the brain's gotten pretty good at. The other thing is that we know that the hypothalamus, which is in the diencephalon, it's not far from this reward site, and it's highly connected to the reward site, has neurons in it that are sensitive to food deprivation, to osmolarity in terms of calculating thirst, oxygen concentration in the blood, salt concentration in the blood. So there's a set of monitors in your hypothalamus that can detect deviations from homeostasis. Homeostasis would be the ideal environment for the cells of your body. Now, I feel like those things are actually fairly well understood, and to a certain extent, they're kind of straightforward in the sense of what's ideal for the cells, and all you have to do is detect perturbations from the ideal state, and then all of a sudden you've got a motivation, and this motivation can be encoded in action potentials and sent to the appropriate place to say, okay, there's water, and you really need water if you're going to survive for another day or two, right? When people's thirst mechanisms go off, it's life-threatening.
Yeah. Again, I can see some parallels to artificial intelligence. As I think about computers and AI, for example. So the brain has set of, it sounds to me at least, Howard, I don't know much about this. It has a set of experience-based heuristics. And because they are in heuristics form, it can just implement it pretty easily, pretty quickly. If it is true, then you can see it's not doing really well if you take the individual to a completely foreign environment, right?
Yes, that is absolutely
the past
true.
experience
That's correct.
is here.
Yeah, I agree with that 100%.
Yeah, so I think that is, in some sense, that's what we're trying to do in AI too, that you can actually, because heuristics can be done or implemented, pretty costlessly. That is what you want to do. We'll take a quick break, Howard. When we come back,
okay,
we'll talk about your recent paper, How
Sounds good.
Patients Influence Pain. Thank you.
This is a Scientific Sense podcast providing unscripted conversations with leading academics and researchers on a variety of topics. If you'd like to sponsor this podcast, please reach out to info@scientificsense.com.
So we're back, Howard. We were talking about pain, how the brain recognizes it, acts related to the stimulus that it's receiving from other parts of the body that might require some action, and it's a complex phenomenon. You have done a lot of work in this area, but it gets even more complicated. You have a paper in 2018 entitled "How Expectations Influence Pain." You said actions of individuals are guided by the motivation to satisfy drives, and these drives through actions provides a benefit in terms of survival and reproductive success. But more importantly, it is really the ex ante expectation that sort of sets the foundation as to how the brain recognizes how much pain there is for something.
Mm-hmm.
So how does it work? I mean, the brain has some sort of a prediction. Is that what's happening?
Yes. So we talked before in the earlier segment about how there's an intrinsic predictive nature to any kind of painful stimulus. It's predictive of tissue damage, and the time course, will be predictive of how much damage there's going to be. If it seems like the stimulus intensity is going up, it's more frightening, and we said that it turns on an amplification system. If the stimulus intensity is falling, it's predictive of pain relief, and there's a top-down inhibition of pain because there's really less of a drive, less of a motivation to respond. Now, in addition to the intrinsic properties of the painful stimulus itself, over time, we come to associate environmental cues, let's say, a doctor's white coat with a needle and a syringe causing pain, or we come to associate a bottle of pain pills with relief of a headache, let's say a couple of aspirin with relief of a headache. So these cues are neutral, right? But they predict that there'll either be a painful experience or there will be pain relief, right? So these neutral cues come to acquire a predictive quality. And it turns out that predictive quality through these same top-down pain modulatory systems can either relieve pain or enhance pain. So we call the pain relief due to expectation. That's what we call a placebo response. So almost everybody has heard of, well, you take a sugar pill and because you think it's going to relieve your pain, it actually relieves your pain. But it hasn't really relieved your pain. Well, the fact is, yes, it actually has relieved your pain. And there's evidence that it has relieved your pain through the same circuit that's activated by morphine, this top-down pain inhibitory system. And this was something, back in the 70s that John Levine and I looked into. The endogenous opioids, endorphins, were discovered in 1975. And we were working in the lab at that time. And we said, well, let's see if there's something going on here, in people. And it turned out that we could block the placebo analgesic effect with naloxone. Naloxone is a drug that blocks the mu opioid receptor, which is the target of morphine, which is the target of endogenous opioids that act to reduce pain when pain relief is predicted. So it looked like, that it was a similar pathway for a drug-induced effect and for an expectation-induced effect, right? So that's, how we came to understand how expectations can actually change what you feel at any given level of tissue damage. And that turns out to be very important. In fact, there was a recent study that I refer to in this 2018 paper out of Canada, where they looked at over 2,000 patients with chronic pain who were entering into a multidisciplinary pain treatment program. And then they were followed up for 6 months, and they were looking for, what is it about these patients that predicted whether they would respond well to whatever the treatment was that was given. And the amazing thing that they found in the study was the best predictor of whether a treatment for chronic pain was effective was whether the patients expected that the treatment would be beneficial when they entered the study, right? So this isn't just, a cute little, psychological phenomenon. This is something that has big, big clinical implications, and it's critical to understand it. And I think we are beginning to have some kind of understanding of this.
Yeah, it puts the placebo effect in a slightly different context, right? So when we think about clinical trials, we have this control group, we have the treatment group, and we try to look at the difference between the two. But if you are saying that the placebo has a systemic effect, it actually creates effects that are very analogous to a drug-induced effect. Then how you conduct clinical trials may need to be rethought, perhaps.
Absolutely. And people are giving this a lot of thought. I think you put your finger on an absolutely critical concept. And failure to understand this has led to the failure of clinical trials in the past. So obviously, one of the things you want to do when you're analyzing your data is you can't just divide the patients into those receiving the active drug and those receiving the placebo. You actually have to divide them up into 4 groups. This is sort of the critical insight. And of the people who get the placebo, they're going to be divided into those who think they got the placebo and those who think they got the active drug, right?
Yeah.
And those who got the active drug are going to be divided into a group that thinks they got the placebo and thinks they got the active drug. So what you have to do is, when you're analyzing your data, match the patients for similar expectations and then analyze the data that way, right? Instead of the typical way, which is you throw, all these variable expectations into one group and into another group. Because if you think about it, the people who got the placebo but think they got the active drug, they're going to have an enhanced effect from the placebo. The people that got the active drug and thought they got the placebo are going to have a less effective, analgesic response, right? So the very nature of clinical trials is going to minimize the actual clinical effect of any drug, right? So you're almost shooting yourself in the foot with the traditional design for pain studies. And I've tried to explain this to, my friends in the pharmaceutical industry, and I just get a blank stare.
This is so fascinating, Howard. So let me see if I understand this. So it's a 2x2 matrix. On the x-axis, you have the placebo arm. On the y-axis, you have the treatment arm. And on the x-axis, you have 2 buckets. One is expected drug. The other is expected placebo. And you have the same on the y.
Exactly.
So you have
exactly
4 cohorts of people, right? 4 cohorts.
Exactly.
Of patients.
That's what I'm saying.
And you want to compare. Let me see if I can understand this, Robert. So you want to compare. So the real effect, real is not the right term, but people who did not expect to get the drug, but got the drug, will be sort of the effect that is devoid of any placebo effect, right?
Correct. And those should be compared to those who got the placebo and thought that they got the placebo, right?
And
so you want to match people for expectation and do your drug placebo comparison within that group for both groups. You can, after you've done that, you can put your data together.
Yeah. I mean, I don't know much about pharmaceuticals, but this sounds so intuitive to me. Why do you think? Why do you think they don't do it this way?
Because they're very traditional, and the FDA says this is the trial that you need to show us, a statistically significant effect of your drug, and if you do that, we approve it, right? And
Right.
you know, because you've been in the pharmaceutical industry, that it's the pharmaceutical industry goes to, what do they call, clinical research organizations, CROs, right?
Yeah.
And that's what they do.
Right. So we have templates, and
Exactly.
we use templates. And this type of a trial also, Howard, might require more patience. So it has an impact on the cost side, I would imagine.
Well, I would argue no, right?
Yeah.
Not necessarily. I mean, if you say that what you're doing is reducing the variability, then you wouldn't need a big cohort. You could reduce the variability with the same number of patients, right? And you can pool your results from the two groups.
That is true. Yeah. And like you said, it could also make some trials turn out to be positive. That is marginal today.
That is
right. And it might actually show effect.
Right. Yeah. And it also could be, so here's another thing to possibly consider, is that for those people who are expecting to have a drug effect, their central nervous system is going to be operating differently from those who think that they're getting the placebo, right? And if you're saying, well, I've got a drug that's acting on the nervous system, the effect of the drug may depend on the state of the nervous system when the drug enters the brain, right?
Yeah.
So you could say, in the clinical situation in general, a doctor comes in, they've got gray hair, they're wearing a white coat, they have a nice voice, they're friendly, they've taken their time, to give you a thorough examination. They say, hey, look, here's this new drug. I've had a lot of good responses to it. I think this is really going to help you. And then you give the drug. And I'm saying that in the clinical situation, you're going to have a bigger drug effect. And that's why you need to, show that expectation has this big effect. And then the other thing that I always tell all my friends who are taking care of pain patients is, you need to actually ask people what they expect is going to happen, from your treatment, right? Because a lot of times people will say, look, doc, I've tried everything, nothing's worked, right? They're coming to this referral center, and I'll say, well, do you think I can help you? They'll say, well, I hope so. No, I said, do you think I can help you? I said, well, I don't think so. And then I say, well, why are you here? He said, well, because, my uncle told me I absolutely had to come to UCSF.
Yeah, that is what I have a bit of a worry, Howard, that going back to the clinical trial design, there could be some personality and behavior biases in those buckets.
There have to be. That's true.
And so I wondered if it kind of bleeds into the statistical observations. I haven't really thought about it, but
how could it not? It has to, doesn't it?
It has to, I would think. Yeah. So you say, some people are going to think that, let's call them optimistic. They got the drug. They're going to have good effect. They're going to get over it. Some people are always going to think that they got the sugar pill. Nothing is going to happen.
Well,
right?
all I'm saying is that you can reduce the variability within a population by matching people who have similar expectations, right?
Yes. Yeah. Now, the question would be the way they are wired has any bias.
Oh, sure. That's true. That's a really good point. I would say yes to that. And then you go back to something you said earlier in the segment is, everybody's different.
Everybody is that I think, like you mentioned, at some level you could standardize. But if it is the wiring diagram itself has some biases, then we will always find some type of people in those buckets, populating those buckets. And but the idea is a good one, Howard. I mean, it's not just for pain; it's pretty much for any clinical trial, I would think.
Well,
right?
I would say any clinical trial where your outcome is a subjective measure, right?
Yeah.
So for depression, for anxiety, for pain, for itch, a lot of things there. I mean, if you're using an objective measure, so for example, let's say you're looking at breast cancer and you've got a new chemotherapy, you can do imaging studies and show that your chemical actually reduces the size of the tumor over a period of time. That has nothing to do with the patient's subjective experience.
But don't we see placebo effects even there?
That has not been possible to show. I looked for that intensively when we were doing this placebo research, and really, the effects of placebos on objective measures, it's quite minimal. There are some papers that show improvements in wound healing with experimental, wounds.
Yeah.
But that's about it from what I'd seen.
Yeah. So maybe it is something to do with the brain. So if it is a CNS type issue, there are decisions being made in the brain. There are different circuits involved, and the brain is sort of modulating between the circuits. And so you would think maybe it is a CNS type issues where this could be affected.
That's where I put my money.
Yeah.
And there's evidence from functional imaging that it is specific places in the CNS that are associated with, placebo responding. Number one, you get a reduction in the neurological pain signature for any given intensity of pain. So that for sure happens. But then there's another set of circuits that we generally consider to be involved in the decision-making process. These are activated, and correlated with placebo responses.
Right. I want to conclude with your paper on opioid. So it is entitled Understanding Opioid Reward. You say opioids are the most potent analgesic in clinical use. However, their powerful rewarding properties can lead to addiction. And the scientific challenge is to retain analgesic potency while limiting the development of tolerance, dependence, and addiction. Do we have a way to do that?
There are people that are looking into this for a variety of measures. There do seem to be different chemicals that are more or less safe. So one of the more interesting drugs that we have now, it's a drug called Buprenorphine, and it acts at the mu receptor. It has some other actions at other receptors, but it's what's called a partial agonist, which turns out to make it a safer opioid. So for example, for any degree of pain relief with buprenorphine, let's say compared to morphine, it produces less respiratory depression. And it's respiratory depression is what makes opiates like morphine, like fentanyl, like oxycodone or OxyContin, heroin, that's what makes them dangerous. It's the respiratory depression. So it suggests that there may be a way to develop new compounds that cause less respiratory depression. Now, in terms of whether there's a difference in how rewarding a drug is for any degree of pain relief, that's a more difficult challenge. There are drugs that are not opioids The most common, of course, is aspirin or ibuprofen, right? Which, some people call Advil or, whatever.
Yeah.
And if you don't have pain, they're not rewarding, right? So nobody takes, aspirin to get high, right? But you could have a very bad headache and an aspirin will relieve that headache completely. So under certain circumstances, you have these drugs. So what that says is, at least in theory, you can dissociate pain relief from addiction.
Yeah. Now, can we view addiction as related to this prediction the brain has? So if you sort of do this over time, you are conditioning. So the brain predicts an effect, it doesn't quite get it, and so it wants more. That is what's going on. It's not really satisfying its expectations.
Yeah, well, there are two parts to it, and this gets into how do you learn, for example, to associate things in the environment with either reward or pain, right? The way that happens is you do something, and if you feel good after you've done it, then you're going to do it again. If you feel bad after you've done it, you're less likely to do it again. So that would be aversion, right? So we know that if you, say, inject an opioid, it's going to make you feel good. That's going to make you want to do it again, right? So over time, with repeated self-administration of a drug, you learn, that, going to the dealer, buying the drug, getting the hypodermic, injecting yourself, you go through all those motions, you get high, you feel good, so you're more likely to do it again. But there's another part to it that's seen with opiates, and that is if you're taking them regularly and you stop, you feel badly. Somehow you've changed the homeostatic settings of your brain so that if there's no opioid in your brain, you feel a craving. You feel that you need it, right? And that's called abstinence or withdrawal. And that in itself creates a motivational state that makes you want to have the drug. Very similar to being food deprived, feeling hungry, and that increases your motivation to go out and look for food, buy it, and take it home and eat it, right? So there's a motivational part, there's a learning part to addiction. Both seem to a certain extent to involve this reward pathway that somehow when you're in this abstinent state, your reward pathway is suppressed and it makes you feel the hunger, the craving. So it could be that some of the same circuits in your brain that are acted upon by opioids are involved both in the learning and in the craving, both in the motivation and in the reward, that you feel when you get the drug after you've craved it.
And the tolerance aspect of it, Howard. So alcohol tolerance, for example, is it because the circuits are getting less sensitive? So the utility that you expected with the dose, you don't quite get it. So you have to increase the dose.
Correct. That's partly related to it, but tolerance also has an independent component to it. And the general feeling now is that there's one of the major mechanisms for tolerance is an uncoupling between the binding event of, let's say, morphine to the mu receptor. You get a certain amount of binding of the drug to the receptor, but the receptor signals less in the cell, right? So there's a uncoupling of the drug from the cellular effect of the drug. That is basic. That's what you would call tolerance, pure and simple. Same drug, less effect. Dependence is somewhat different. Dependence is defined as an unpleasant state that occurs after you've had the drug, right? And then you stop, right? That, for example, you would never get a withdrawal state from an individual who'd never been on an opioid. It just doesn't happen. You have to be on it for a certain period of time and at a certain dose. The higher the dose, the longer you've been on it, the more severe the craving and the abstinence effect, which can be literally painful. When you stop taking an opioid, it isn't that the effect just goes away, it's that you feel terrible. And part of that feeling is actual aches and pains in your body. It's like a bad flu.
Yeah, it is. It has become a big problem. So Howard, in conclusion, you have done so much work in this area of pain. As you look forward next 5 years, where do you think we could make additional discoveries, innovation? Where is most likely that we will make?
Oh, there's so many places. I mean, number one, we need to have better understanding of how the peripheral nervous system senses tissue damage. There's a lot of work being done on that in many labs. What is it about injuring a nerve that makes it become painful? How does it start generating new pain signals where there weren't signals before? So there's many projects going on to help us understand the disease process in the body. There's another, completely separate field of what are the changes that occur in the central nervous system when there's persistent pain, right? So the big problem is not how do you treat acute pain. We're very good at that. Millions of people are having surgery every day. It's painless because you're anesthetized, and postoperatively, we can control your pain through a variety of methods. So I would say acute pain, we're good at that. Chronic pain is really the problem. And there's general consensus that this occurs through changes in the central nervous system due to a persistent painful input, right? So what are those? Now there's no general agreement, but there are multiple independent lines. People are looking at the spinal cord, they're looking at the peripheral nervous system, they're looking in the central nervous system. The thing that's most interesting to me is, what's the circuitry for expectation? And are there ways that we can intervene in the central nervous system to tap into those circuits that either facilitate or inhibit pain? I expect to see a lot of progress along those lines. And finally, I think we're going to be able to do a much better job designing pain relievers that precisely target, the pain suppression mechanisms in the brain or the pain amplification systems in the brain, right? There's already preliminary evidence that, there are modifications of the receptor. These can be targeted, using, I would say almost you'd have to use supercomputers because we have the crystal structure of these opioid receptors. We can use our understanding of that structure to determine which of the current drugs that we have are the best and how can we improve on them, which by using pharmaceutical chemistry. So I would say a combination of, crystal structure, computational chemistry, medicinal chemistry, pharmaceutical chemistry, and clinical observation make me very optimistic that over the next decade or two, we're going to have some much better ways to treat our patients with both acute and chronic pain.
Just a quick question also, Howard. So do you think we get closer to sort of simulating what is happening in the brain, the whole CNS system in silicon somehow, so that you can, chronic pain without knowing anything about it? It's almost like a stuck switch, right? It seems like
that's
keeps sending
a
the
great
same
question.
signals.
And I generally tend to be an optimist, but I would say that so far we haven't had the I would say the theoretical aspect, the computational aspect for brain function. We really don't yet understand fully how information is represented in the nervous system. We know about cells, we know about synapses. We can record from lots of different cells, but it's a huge computational problem. You'd have to record from thousands of cells at multiple sites in the brain. We're starting to approach that. Then we have to sort of come up with some theoretical concepts about how the information is represented, in the inhibitions and excitations at the different stages of the circuit function. We'll get there, but we're not there yet. And what I would say is we need a stronger theoretical approach to information processing in the brain. We've got the chemistry, we've got the anatomy, we've got the tools to manipulate the brain, but we need some better ideas if we're going to understand how it works.
Yeah, it is. I sometimes say that God has a sense of humor.
Yeah, I can't remember, but some famous scientist said, I think it was a physicist, that it might have been a physicist who turned into a neuroscientist. He said, if we were smart enough to understand the brain would be too complicated for us to understand it.
That's right. Excellent. Yeah, this has been great.
It was a lot of
Thanks
fun,
so much
Gill.
for spending time with me.
Bye-bye.
Thank you.
This is a Scientific Sense podcast providing unscripted conversations with leading academics and researchers on a variety of topics. If you'd like to sponsor this podcast, please reach out to info@scientificsense.com.